Okay, we're going to just talk a little bit about Meselson and Stahl. There is a great animation on sumanasync.com. If you look under general biology, there's a super animation with some little self-test questions throughout it that explains how they did the experiment. You don't need to know any details of the cesium chloride gradient, but you do need to know the sort of general principles. So, here are these guys, Meselson and Stahl. These guys were working just after Watson and Crick had discovered their double helix model and got the Nobel Prize for working out that DNA was a double helix. And when the model of the double helix was proposed, Watson and Crick had to make sure that their model actually allowed DNA to do its functions. So, yeah, big, nice, stable molecule. It has to be stable. It codes for proteins. It has to be big. And they had this idea that actually the sequence of bases might be unique for each organism and therefore might be the code. So that was all good for that. But also, of course, DNA needs to be passed from cell to cell during mitosis and meiosis, during cell division, and therefore it also needs to replicate. So what Watson and Crick said was, okay, we've got this lovely double-stranded two complementary strands. So one way might be to break the hydrogen bonds. So they were saying, right, okay, we're going to take our DNA, we can break that when it's replicating into two strands, and then we could add in some new nucleotides that we're going to use to make our new DNA molecule with, and they'll join by complementary base pairs. Now that seems very straightforward and very obvious that that might be a way of that DNA replicating. And then they kind of, basked in the glory of the Nobel Prize for a bit and left the work to show that that their model was correct to these guys Meselson and Stahl. Now, one of the sort of jobs of a research scientist is to say, well, what if it's not that? What other ways could that possibly happen? What else could possibly be going on here? And then to devise an experiment to show whether their ideas of what else could be going on is correct, or whether the one that looks most obvious is correct. Because of course it might not have worked like that. So they proposed two other models. So this one is called Semiconservative Replication. So this was Watson and Crick's idea. What they mean by semi-conservative is that when we replicate the DNA, semi of it, is conserved from the previous generation. And Meselson and Stahl said, well, what if it doesn't go like that? What if it doesn't? What if it goes like this? What if it goes like you've got your DNA molecule, and by some mechanism you just make a copy in new nucleotides. You just get some new, your cell just gets some new nucleotides and goes, oh yeah, I need to put them in that order. In which case, we will have conserved our original molecule. And they call that model, so these are just, remember, these are ideas that they're going to test. They don't know at this point which one's right. So they call that conservative. And then they thought, well, what if it doesn't happen like that at all? What if it doesn't, what if somehow, and there's plenty of evidence of this going on in nature, butterflies do this, they kind of break themselves up into lots of little bits in the chrysalis, and then kind of reassemble themselves. So They call this model dispersive, so breaking up into little bits and then reassembling some old and some new nucleotides. And I'm not going to draw this because it's too tricky, but if you disperse this molecule and then fill in your new bits and you're making one molecule into two, you can fairly well assume that 50% of your bases, your nucleotides, will be old, your conserved ones, and 50% will be new. It's just they won't be nice and organised into two separate polynucleotides. So, I'm just going to take this down one more generation just to remind you of what's going on here. So, if we went down another generation, remember, we're going to split our strands apart down the hydrogen bonds, break the hydrogen bonds, make it into two strands. So this one will go into a blue strand and an orange strand, and this one will go into a blue strand and an orange strand. Sorry, they're getting less and less double helixy, aren't they? And then all your new nucleotides are going to come in. So now we'd have two that are sort of, we've still got our blue ones from the beginning passed on to the next generation, but now we've got ones that are completely new that have nothing of this molecule in them. With conservative, if we did the same, we'd have our original molecule and we make another copy, and then with our new molecule, this is of course in a different cell, we'd have that and we'd make another copy. And of course in dispersive, which again I'm not going to draw, if we did that again, we're now spreading out our blue nucleotides, our parent ones, into 4 different cells. So we could kind of expect that each one would have 25%, and then of course all the rest of it would have to be made of our new nucleotides. So they then went on to do an experiment. Now you can't colour in DNA, it's too tiny, you can't see it. They couldn't see this happening, so they had to have some way of distinguishing between old nucleotides and new nucleotides. So what they did was they said, right, okay, so we know we can separate out things by mass by a technique called centrifugation. We've got these DNA molecules in cells. We need an organism that will divide at a pretty constant rate. So they chose E. coli, a bacteria, divides every 20 minutes. So, if you got your parent molecule and you left it 20 minutes, you'd have twice as many cells and the DNA would all have replicated. And you can leave it another 20 minutes and the DNA all replicates again. And you can take your samples out. So, separating things out by mass, I thought, right, okay, so if we label our original DNA molecules with a heavy nitrogen, then all our nitrogenous bases we can label up and those will be heavier. So that's exactly what they did, and they did, as you can see on the video, a little experiment to show that actually you can do that and that does work. So they had heavy and light DNA and they could separate them out, and they thought, right, jobs are good and now we can start. So in the centrifuge tube, so this is just spinning round and anything heavy will go to the bottom. I'm just going to mark on my centrifuge tube 3 bands. And again, on that animation you can watch that spinning happening and where the DNA goes to. And you're just going to have to take it on faith. It spins around a bit like a fairground ride. And you know how you get flung out sideways if you're being spun, or if you've done the I'm going to spin round with a bucket full of water and none of the water falls out. Same principle. So, what they found was that the heavy labelled stuff falls to the bottom. And they called that generation 0. So this is generation 0, and in all three, in the tubes, we would expect in all three that because we've got heavy DNA at the top, that it would all sink to the bottom. When we get to generation 1, and again, we're doing a model for all three tubes, and then we'll look at what they actually got. Got a top, middle, bottom bands in each of the three tubes. What we would expect in this is that each DNA molecule is kind of half heavy, half light. So it won't go to the heavy position because we've got some light nuclei, and half of the molecule is light nucleotides. It won't go to the top because we've still got some heavy ones, so it'll land up in the middle of the tube. With the conservative, this one will still be heavy and will still go to the bottom of the tube, so any cells with the labelled nitrogen in will go to the bottom, and the DNA from the cells that have got the new DNA molecules in, they'll go to the top because they haven't got any of these heavy nitrogen isotopes in. If we look at the dispersive model, we said it doesn't really, I'm not drawing it because it's too complicated, but we were saying we were splitting that DNA molecule between two new molecules, and we would expect that each of them would have 50% of the heavy and 50% of the light. A bit like the semi-conservative one, it would just be a bit all over the place. So again, that's going to go to the middle. All those molecules will go to the middle. Now that generation immediately doesn't allow you to distinguish between these two that are both in the middle, but it would, when you got your results, allow you to either eliminate or say, oh yeah, that's the one. In generation 2 then, so again remember we're just modelling now, we're going, yeah, this is what would happen, these are the results that we would expect for each of these three models. My test tubes are getting more and more different. So, in this one, we're now in this generation, so we've still got some that are sort of half heavy, half light, and they'll be in the middle, but these ones have got no heavy nucleotides in at all, so they'll be up there at the top. In this model, we've still got our heavy DNA preserved, and we've now got more at the top. So that band would get thicker. And here, each of the molecules has some heavy nucleotides in, but now, where we started off with 50%, we've had to now divide it between another two cells, so we've only got 25%, but every single molecule will be the same, so we'll form one band, and it will be kind of halfway between. So, those are the hypothetical results. So what did they actually get? So, when they actually did this experiment, the results they actually got, their tubes look like that. So, how did they know then that their other models were wrong? Well, if we look at generation 1, this is a half and half model, and this one isn't. So if you don't get that, it's not conservative. So this generation shows that doesn't happen. And it suggests that your replicated DNA contains half of the original DNA molecule and half some new nucleotides. Doesn't tell you whether it's dispersive or whether it's been broken down the middle, but it does suggest that this means that, so that tells us that half of our new molecule contains old, I'm going to put that in brackets, old nucleotides. Then go down to the next generation. Now we've already said, right, okay, that doesn't happen. And in our next generation, in semi-conservative, we've got 2 bands, but here we've only got 1. So this generation here, the second generation, shows us that it's not dispersive. So this tells us that the replication is semi and that old nucleotides are preserved as a polynucleotide strand. Now, you need to be able to, in an exam, you need to be able to define semi-conservative replication. You need to know how it works. You need to be able to sketch this kind of diagram and place the lines on the tube. So they could say, in the next generation, where will the lines be? Now, clearly, in the next generation, you're still going to have that band in the middle. That band in the middle are these hybrid ones where the old heavy nucleotides are preserved. So every time you split one of these hybrid molecules, you're going to get another one, and you're going to get more and more of these. So this band at the top will get thicker and thicker and thicker. So you might be asked to do that. But let's just have a nice definition of semi-conservative replication. So these are the 2 guys that show that this is how it's done. There's kind of 2 bits of this definition. There's one that's sort of how it happens. So each strand of the parent molecule is used as a template for new nucleotides to attach by complementary base pairs. So that's the how, and the results is that each new DNA molecule, and it has to be a molecule, you have to really know the difference between whether you're talking about a single polynucleotide strand or the molecule made of 2. Each new DNA molecule consists of 1, I'm going to put that parental template strand, and 1 newly synthesized. That's important. You can't just say one new strand. One newly synthesized strand. So don't just say old and new. Do say parental or template strand and newly synthesized strand. So Meselson and Stahl, that's all they did. They proved that Watson and Crick had been right in their original idea. This is how they did it. There's loads of information out there. I think the textbook does a really good job of explaining how the experiment worked. So, good luck.